all eukaryotic cells contain actin and microtubule, but not all organisms contain intermediate filaments, which is a major type of cytoskeletal element. This type of filament or this type of cytoskeletal element is primarily found in metazoans. Metazoans are multicellular organisms which have bodies made up of different types of cells. This, for example, includes humans, nematodes, and mollusks, which are snails, oysters, for example. They do have intermediate filaments, but single-celled organisms, they do not, even though they are eukaryotic. So let's talk about intermediate filaments. Intermediate filaments are not required in every cell type. Even in advanced organisms as our cells, the specialized glial cells, which are also called oligodendrocytes, which make myelin in vertebrate central nervous system, they do not contain intermediate filaments. Cytoplasmic intermediate filaments are closely related to their ancestors that are much more widely used. For example, nuclear lamins that we have talked about previously. These intermediate filaments are related evolutionarily to the nuclear lamins. During metazoan evolution, lamin genes have duplicated and evolved to produce rope-like cytoplasmic intermediate filaments. The nuclear lamins, as you may remember, are proteins that form the meshwork lining the inner membrane of nuclear envelope, where they provide anchorage sites for chromosomes and nuclear pores. If the mechanical properties, intermediate filament mechanical properties are useful to soft-bodied animals such as nematodes and vertebrates which do not have an exoskeleton, like for example insects, crustaceans, they have an exoskeleton, hard skeleton on the surface of their body. They're soft inside. So the animals that have softer outer parts, they require this type of strength in the tissue, which is provided by intermediate filaments. Intermediate filaments are prominent in cells subjected to mechanical stress, where they provide physical strength to the cells and tissue. For example, we have talked about, when we were talking about skin, we said that epithelial cells in the skin, they're linked together through these intermediate filaments via adhesion junctions called desmosomes. Intermediate filament polypeptides are elongated molecules with an extended central helical domain that forms a parallel coil with another monomer. A pair of parallel dimers then associate in anti-parallel fashion to form a staggered tetramer. I'll show you this in the next slide, a diagram of this, what I mean. But I would like to point out here that this tetramer basically is the soluble subunit of intermediate filaments and is analogous to alpha-beta tubulin dimer or actin monomer. Now let me show you structure of this monomer, intermediate filament polypeptide. First of all, let's see, this is the alpha helical polypeptide monomer of intermediate filament. This is the alpha helical region. This is the amino terminus, and this is the carboxy terminus. Two of these come together. They associate with each other in a parallel fashion. What do I mean by parallel fashion? The amino terminus of this protein and its partner protein in the same location. So amino terminus of one protein and amino terminus of the other protein, intermediate monomer, they are lined up in such a way that they are parallel to each other. This dimer then associates with another dimer in an anti-parallel fashion. It is reverse of parallel. So here there is a amino termini of those dimers that we formed in this previous part of the slide. The amino terminus are adjacent to the carboxy terminus of the other partner dimer. So, and also, not only they are anti-parallel, they are also staggered. Staggered means one dimer starts here and ends here. The other dimer does not start at the same part where the previous dimer was starting. it is starting somewhere in the middle of this dimer. So this is anti-parallel orientation. The dimers associate with each other in anti-parallel fashion, whereas two monomers associate with each other in parallel fashion. Here, I would like to again show you that this is staggered. What the significance of the staggering is that when these molecules are staggered, the next one will probably come somewhere here and extend all the way here, and the next one will start somewhere here and go all the way here. So this basically is this association again is through weak interactions, the hydrophobic bonding, which is keeping these subunits together, these monomers and these dimers together. And as you put the new ones on, you are staggering them. This basically results in formation of a long filament which has staggered subunits, staggered parts, which ultimately gives it the tensile strength, which is the function of intermediate filaments. In the cross section, you will see a cross section of intermediate filament, which is 10 nanometers. You will see that they have 32 individual alpha helices, and these, of course, as we have seen, that alpha helices come together. These 32 are in form of pairs, which means there are 16 dimers or 8 tetramers. Let me show you an EM, electron micrograph of intermediate filaments. You can see these long rope-like structures. They have great tensile strength and they are flexible. Please note that this filament is, for example, bending here. This is a very acute bend here. So these filaments are well suited to maintain cell integrity because they are flexible, and they are their strength is great because they're staggered when they line up with each other. Let me show you a graph. Here we took the way this experiment was done is that microtubules, actin filaments, and intermediate filaments, they were all put in our experimental tube in equal concentration, and they were exposed to a shearing force in a viscometer, and the resulting degree of stretch was measured, and here's what the results show. Microtubules are easily deformed. This is the amount of deformation. So microtubules can deform even when small amount of deforming force is applied. So this is amount of force being applied in the x-axis, and the amount of deformation is on the y-axis. So even with a small force, microtubules can deform easily, but they also rupture. They cannot take too much of this deforming force beyond the 150%, for example, of their original length. Actin networks are more rigid. They do not deform as much, but they can take more amount of deforming force. However, they do not deform easily. They are more rigid, but they also rupture at a certain point. Intermediate filament networks are easily deformed, and they can take a larger amount of deforming force without rupturing. Protein phosphorylation regulates intermediate filament disassembly. So if you have something that's strong, we have seen that cells are dynamic entities. They need to reorganize their shape. Sometimes they have to reorganize their shape. They have to reorganize their cytoplasm. So nothing should be permanent. So how you get rid of intermediate filaments? The same way you got rid of nuclear lamins is by phosphorylation. So if you want to get rid of intermediate filaments, you just phosphorylate and they will fall apart. Labeled subunits, if labeled subunits are injected into cells, these will incorporate into existing intermediate filaments within few minutes. So they are dynamic. They do exchange their subunits. If we, however, inject polypeptides just with the conserved helical domains without their carboxy or amino termini, they act as dominant negative, and they will basically disassemble the intermediate filament network. Not only that, they will also have a substantial effect on the other cytoskeletal elements, microtubules and actins. Actin filaments, they will also disassemble. This goes to show that these three types of cytoskeletal elements, they have a combined effect. They can cross-talk with each other, and things that affect one can also affect the other type of cytoskeletal element.